All right, thanks everyone for coming. I'm really excited to introduce Bofu Zheng. So Bofu and I met at a couple of conferences ago. He was still a PhD student at his script working with Drew Lucas. And one of the exciting things I think about his field of research is it's really at the intersection of physics and biology. So a lot of biophysical interactions. So you'll see today, right? Strong PO person who met Bofu at a physical oceanography conference and came in and taught me a lot about the impact of the physics on the coastal circulation. In this case, we're talking about red tide. So and Bofu is now a postdoc at HUI, again working on an intersection of biophysical interactions, and he'll talk a little bit about what he's doing right now, so that if there are interested parties for semi-local collaboration, please come to lunch afterwards. We can talk more. All right, Bofu, go ahead.
Thank you, Jack. Yeah. Okay, thank you everyone for coming. And before I start my talk, I just want to say thank you, Jack. Thank you a lot of people, Mary, Lauren, and all people for these organizations. And then I'm very happy to be here today with this talk regarding this my thesis work. And then hopefully to convince you and tell you guys a story where it shows the really powerful combinations of different types of observational tools that really enables the observations of this dinoflagellate. They are able to vertically migrate from the surface into the deep nutrient pool and collect the nutrient. And then that's, we believe, as the primary reason for they are able to achieve a really huge biomass during this red tide event that happened in Southern California in 2020. So again, the whole story is to show the combination of the physiological behavior of dinoflagellate with their metabolic activities. So this work has recently been published in PNAS. So if you are interested in more details, so feel free to check it out. And then before I dive into too much detail for the story, I want to acknowledge my collaborators, who are Drew as my PhD advisor at Scripps, and also Peter, Tamara, Clarissa, and Wei from Scripps, and Kristen Davis from UC Irvine, and also Heidi Sosik from HUI. So the funding for this project comes from the schools program, which I'm really grateful for. Then let's get started. So the talk will be in 4 different parts. So I start with the introduction. So again, so what are half-algal blooms or HABs? So basically, HABs are like accumulation of phytoplankton, a huge amount of phytoplankton in a short amount of time. They will sometimes discolor the ocean, as you can see from that image. To the right shows the whole surface color of the whole surface of the ocean is printed in yellow. So sometimes they are called a yellow tide, and they were painted in red, so they are often referred to as the red tide. So you can see they are also very patchy and very predominant in the coastal ocean. And then HABs, the reason why we want to study HABs is mainly because HABs can have the negative impact on a lot of things, including like human health, because some HABs species they produce toxin, and also due to this negative impact, it can cut like huge dramatic loss on a lot of economic stuff. For example, like people need to spend a lot of money on how to control their pollution and also try to reconstruct the ecosystem function after the HAB dies. So here are some images of the negative impact to the HABs. You can see all these dead fishes once the bloom begins to decay, and also these smelly foams like floating on the ocean surface. So you can even smell this very like at least very bad to me. It smells like miles away from the coast. And you can also see from this image that the beach are close. We don't have very any fun, like at least for me during the red tide event. So therefore we need to have a better understanding of the HABs, including their initiation, their maintenance, and also their propagation. So one of the topics that I focus on for this project is mainly in the initiation stage of the bloom, which I will dive a little bit further. But before I dive into further, I want to just show this introduction. So the HABs can be dominated by multiple different species. So especially for this dinoflagellate species, I believe hopefully people have some is familiar with the dinoflagellate. So they are like a kind of marine phytoplanktons. So they have really interesting and key trait or aspect, which means that they can vertical migration, they can swim. So here I'm showing a result from a not recent paper, but a really clear map of the chlorophyll concentration as a function of density in the y-axis and also as a time. So you can very see a clear time evolution of the cross-isopycnal signatures of these patches of chlorophyll concentration, which is a strong indication that they were swimming across isopycnal in this time perspective. Another thing that I want to emphasize on, another evidence is that this is actually dates back to around half a century ago, like back to 1976, 57, where a lot of researchers, like at least these authors, they found that during a HAB event or during a red tide event, the near surface nitrogen is normally pretty low. So there's a contradict between how the huge biomass that you observe in the red tide with the low nitrate concentration near the surface. So therefore, people were wondering, the researchers, they were wondering, what is the nitrogen source in order to support this huge biomass as you observe in the red tide. So a really fantastic hypothesis was brought up by Atmospheres trying to link this vertical migration ability of dinoflagellate with the low nitrogen that you see from the surface, which is this but vertical migration hypothesis. So this was brought up by Atmospheres et al. In their 1968 paper, which is around half a decade ago, where I just want to show a really quick animation to illustrate this hypothesis. Where the hypothesis that these dinoflagellates, which is this little guy here, they will vertically migrate downward from the surface into the deep nutrient pool during nighttime, as you can see this animation. And then once they were down there, they assimilate nitrate, because normally nitrate is elevated near the subsurface, especially lower down portion of the water column. And then once the next day, when the sun comes up, they will vertically migrate upward from the deep nitrate pool back to the surface, and then photosynthesize. So therefore, with this combination of vertical migration and also the nitrate assimilation ability, they are able to both accumulate enough nitrate for them to grow or to dominate during the red tide, but also photosynthesize to convert this nitrate into energy, and then for them to sustain during the red tide. So yeah, it's a pretty smart hypothesis, and a lot of experiment actually, mainly in the lab, has been done to understand what is the petroleum mechanism of the vertical migration of the dinoflagellate, and then what is how the environmental factors, for example, light, nutrient concentration, and temperature modulate their migration behaviors. However, there's a really important aspect that this hypothesis has never been really tested in the field, like in other words, with real in situ observational oceanic data. So this is actually the knowledge gap of this project, is to try to test this hypothesis with the in situ real oceanic data. But the challenges, you may be wondering, like, why nobody has tested hypothesis over half a century. So this is actually mainly comes from the difficulties in the in situ observational world. So think about that. If you want to test this hypothesis, first you need to collect high resolution profiles or time depth map of the chlorophyll concentration, physical oceanographic concentration, physical oceanographic data, for example, temperature salinity, as well as nitrate, like these biogeochemical data, like nitrate concentration. But people normally do with their nitrate concentration measurement is to collect water samples subsurface, and then using these Niskin bottles or CT represent, and then bring them back to the ship, and then put them into like little bottles, and then bring them back to lab to analyze their nitrate concentration. So with this combination of these Niskin bottles, lab measurements, we are really hard to measure nitrate concentration with both high resolution in space and time. So here I mean vertical, like space resolution, in order to test this hypothesis should be like less than 1 meter or even less, and then temporal resolution maybe on the order of like 20 minutes or even like less than 1 hour. So therefore, it's really hard to use this combination to be observing this diel vertical hypothesis or test this hypothesis in the real field. So therefore, we need some technology breakthroughs in order for us to collect high temporal and spatial resolution data in order for us to test this hypothesis. So here I'm going to dive into some technology breakthroughs that I used over my PhD, especially for this project. So the first thing is called the wire worker. So I'm going to show a slow motion video of nothing here. typically there should be a video of the variable package, but no one's, yeah, I'll probably just show this first. So basically, the wire is an underwater profiling system that consists of the surface buoy. So imagine that there's a surface buoy here, and then the wire that's suspended from the surface buoy to the bottom anchor, and then this variable body, which is this yellow package, spanning along the, yes. Yes. Yeah, I think I probably just describe this backwards. Sorry, yeah.
It might come up.
It might come up, yeah, yeah. But basically, you have a variable body that is this yellow package here. Imagine that there's a yellow package that's stand along this wire and moves up and down along this wire and like the whole water column, physical and biogeochemical data. So the wire place has really some really good advantages. For example, it only uses the surface wave power. So in terms of the mechanism of the motion of the wire is that it uses the relative velocity between the wave velocity and then the variable velocity. So therefore, imagine that if this is the wire and this is the variable body, so when there's a relative upward motion between the wire and the wire curve, the cam inside the wire curve will disengage the wire curve from the wire. So therefore the wire curve will slide down along the wire a little bit during the upward phase of the wave. However, when there's a downward relative motion between the wire and the wire curve, the cam inside the wire curve will grab the wire and stop the wire curve from sliding up. So therefore, within each surface wave cycle, the wire curve will slide down along the wire a little bit. And once it reaches the bottom anchor, there's a trigger that triggers like fully disengage the cam of the wire curve from the wire. And then due to the positive buoyancy of the wire body, it will float back from the surface from the subsurface to the surface and complete one cycle. And then the wire will do this cycling like once again and collect the whole water column profiles. And then it's a really fast speed because it only uses surface wave energy. So for normal like 100-meter profile, it only takes around 10 minutes to complete this whole profile. And then if you mount multiple sensors on it, for example, this is a CTD, and this is a optical irradiance sensor. This is an optical backscatter sensor, where you measure chlorophyll concentration, optical backscatter. So when you mount multiple sensors, especially high-resolution sensors onto the wire platform, you are able to achieve a collection of the high vertical resolution. I'm talking about the vertical resolution on the order of like centimeter scale of physical and biogeochemical problems. So this so it's a really great tool for us to study this fine scale dynamics, or in other words, the dynamics that evolve in the ocean with a less than 1 meter vertical scale and a temporal scale of like several minutes or even like on the order of like hour scales. So that's the wire. I'm sorry for this animation not showing here, but feel free to check it out. It's a really cool instrumentation that was developed at Scripps by my advisor and also his advisor at Scripps. Another technology that I want to highlight is this nitrate sensor, which is called submersible ultraviolet nitrate analyzer. So which is this sensor here, this cylindrical sensor that I marked that is yellow oval here. So this nitrate sensor is commercially available by Seaver. So it actually measures high temporal resolution nitrate data on the order of like 1 Hz. So basically this is a sampling scale. So basically you pump water into the sampling region, and then based on the optical like light absorption of the seawater in the UV spectrum, and then this instrument will estimate nitrate concentration. So due to this high temporal resolution, we are able to use this SUNA and wire combination and collect high vertical resolution profiles of nitrate concentration, which I will show just in a bit. And the third technology breakthrough is this thing called imaging flow cytopole, or IFCP. So for people who are not familiar with this instrument, so it's basically an underwater microscope. So you can imagine that you bring a microscope down into the water column, and then this instrument will stay in the water column and collect high temporal spatial resolution of underwater like phytoplankton images on the scale of like ranging from 5 microns to 180 microns. So basically, it really enables the collection of the images, like actually thousands of images within like 5 millimole of the sample that you took, and then started this not only quantify their amounts like this microplankton and mesoplankton account, but also classify their by what they are and what's the portion of the in these samples. So here's only like a very simple image of the thousands of image that the IFCD is able to collect within several hours of time. So cool. So with all these technologies, then we can actually choose or dive into a red hat and start it and test it to see whether these dinoflagellates they are really able to vertical migrate, and also whether they are able to collect or assimilate nitrate from the subsurface. So then let me introduce this 2020 red hat in the Southern California band. So here, it's supposed to be red, but it's looking like a little greenish. But basically, I'm showing a sea surface chlorophyll map of this red hat in the Southern California band. So basically, this is San Diego is down here, and this is more like Los Angeles, and this is this point section. So basically, you can see that there's an elevated chlorophyll concentration along the Southern California band with a chlorophyll concentration of higher than like 100 microgram per meter cube. So it's so just a little bit backwards that normally the chlorophyll concentration in this area is like 1 microgram per meter cube. This is like 100 times higher than the normal concentration. And then there's some interesting thing regarding this red tide in the Southern California is that this red tide lasted for 2 months, so which is much longer than the normal red tide events, which is normally take like weeks. This is like several times longer in duration, and also with a large spatial coverage, which covers the entire Southern California down to the Baja California, which a lot of data that I'm now showing here in the Mexico region. And then the most surprising thing regarding this red tide is that this is actually what we believe is the highest or the most concentrated red tide that's ever been recorded over 40 years of record at the Scripps Pier. So at the peak of this bloom, the pier group measured 9 million cells per liter of this dominant species, which I will talk a little bit next, and then with a chlorophyll concentration around 900 microgram per meter cube during the peak of the bloom. So this is like enormous bloom, and it's really provide really great opportunities for us to understand what is this type responses and also what's the initiation mechanism of this red tide. And then before I dive into too much details, I want to show that you can also see this ironish color. The color may not show clearly by the projector, but there's like ironish color of the ocean due to this red tide. And also we are lucky to deploy a wild mooring, like right close this dormant mooring, not far away from the Scripps campus, right on the 100-meter ATIBAS. So basically on the wild as you can see our payload, we have CTD, we have optical sensors, which measures chlorophyll concentration of the backscatter, and also lag sensors, which measures irradiance, and also sonar, which measures nitrate concentration. And then besides the wirewalker, there was also a long-term ecological mooring called Delma mooring, which gives us velocity data, and also the IFCD, which gives us like this high resolution of images that really enables us to know what is the dominant species and what's the portion of this dominant species relative to other phytoplankton. And then most importantly, we also have this Calcutta station, which is a long-term ecological station, not far away from from our mooring location, which gives us the climatological situation. Because this Calcutta station have been collecting temperature and nitrate data over more than 70 years. So therefore, with this 70 years of data, we can know or try to understand what is the current climatological situation of nitrate, and also compare this climatological situation with the nitrate concentration during this red tide event to see what is the difference of the red tide can cast onto the local biogeochemical field. You can also see these people were wearing masks during this deployment, but this is right at the beginning of the lockdown, if people still remember. This is a really fun fact as well. So again, as I mentioned, I said about the dominant species. So here is the dominant species of this red tide. It's called Lingulodinium polyedra. So here I'm showing like a lot of images that was collected from the IFCDs. So again, these are only a very, very small subset of the thousands of images that is able to collect during this red tide. So this little guy is like around 40 microns in size, and then they are tectonic dinoflagellates, which means that they can swim. They have this flagella that enables to swim, and also they are pretty common in the Southern California and has been recorded of causing red tide in the local water like ever back to 100 years ago. So you can see that these are the images, and then I want to dive a little bit deeper, but before diving deeper, I want to show you this animation before I do say any attention. This is actually a fun fact. During this red tide, you can see this spectacular bioluminescence, like along the San Diego coast, and also even along the entire Southern California coast, with this breaking of the surface waves, and then the emitting of this spectacular like bioluminescence during this red tide. And it's actually fun fact is that people were during like a lockdown there at that time. As you can see, like there are clusters of people standing along the coast to see this beautiful light show during this red tide. Cool. Then let's dive into a little bit about the results and discussions. So before I dive into results, I want to try to show this fact that this whole red tide was actually dominated by this algal. So here I'm showing like 1 times 2 time series. So the first panel or the upper panel is the time series of the planktonic biomass in blue, and also the alcoholic biomass in black. So you can see there's a clear increase of the biomass towards the end of April, and then gradually decrease afterwards. And then with a biomass concentration around like 5,000 micrograms of carbon per liter, which is a huge value. And then the second panel, I'm showing the portion of the biomass between alcoholic and then the total biomass that is able to observe during by the IFCD. You can see that the almost 80% or 90% of the whole water column biomass was dominated by this alcoholic, which is a strong evidence saying that the most like this is a more like a unique species like dinoflagellate that is able to dominate this whole red tide. So yeah, and then the next thing is that I will only focus on this small time period because that's the time period when we deploy the wire. So here I'm showing the wire data with 4 different panels, with the temperature data in the first panel, salinity data in the second panel, chlorophyll concentration in the third panel, and also nitrate data in the last panel. And they are all plotted as a function of depth and time. So the depth is around from surface to 15-meter depth, and time is around 1 week of time. So the green lines contour on the isopycnums. So again, there are many interesting signals inside these 4 panels, which I won't dive too much details. I just want to highlight several key points. The first thing is that you can see this vertical heating of the isopycnums due to the internal waves. And another thing that I want to highlight is that there's a clear offset between the thermocline. As you can see that the surface, the water column during this red tide was strongly stratified, especially temperature stratified. You can see there's a very shallow, less than 10 meters of the very high temperature water, like larger than like 24 degrees C, like occupying this surface region. And then there's a clear spatial or vertical offset between the thermocline and also the nitric line. So the nitric line, by definition, is the place, is the depth where we can see some increase in nitric concentration, and then that's approximately by this line, as you can see. And then the most important thing I want to point out is in this chlorophyll panel, which you can see that there seems to be some coherent cross-isopycnal patches moving down from the surface down into the deeper part of the water column. So you may wonder, like, what are these patterns? Are these alcoholic vertical migration pattern? Because they do seem to be cross-isopycnal with some sort of coherency and also some pattern. But in order to show these cross-isopycnal signatures, the first thing is to decouple this vertical heaving of the isopycnals from this data set. So therefore, what we did next is that we actually map all this data from the text coordinate system onto the isopycnal coordinate system. So basically, you can think about, so I stretch all these curvy isopycnal into a straight line, and then map all this data onto the isopycnal. So therefore, in this way, we can hopefully see a better cross-isopycnal signatures of these chlorophyll patches. So which this is the plot that I want to show next. Is that so now all these data are plotted onto the isopycnal system. So basically, you can think about that for each row, they are actually the same isopycnal, and all the signal cross different rows are representing their cross isopycnal signatures. But before I dive into too much details, I just want to point out first thing that the first panel I'm showing the time series of the near surface viral measured chlorophyll concentration in blue, and then the IFCD based alcoholic cell concentration in green. So basically, you can see that there is a really good correspondence between the blue line and the green line, which is again indication that the algal is the dominant source of the chlorophyll data that we are able to observe. And then the black line are the part data which I will refer to just in a bit. And then the second panel is the chlorophyll concentration, again in the isopycnal color system. So basically the y-axis is sort of the isopycnals, and then the x-axis is the time. So we are showing exactly the same time period. The only difference is that I map all these data from the depth to isopycnal color system. And then the black lines are isonitrate lines contour out. So basically, right now you can see a much clearer, organized or coherent cross isopycnal signatures of these chlorophyll patterns. So they occur like roughly once per day because each grid point is 1 day. And then here, there's one thing that I want to mention is that this vertical migration, we believe these cross isopycnal signatures are the vertical migration behavior of algal. So they are actually cued by both light and nitrate concentration. What do we mean cued by light is that you can see that every day when the sun goes down, which is correspond to this part data, they start migrating down. So therefore, the light actually initiated their vertical migration. And then in terms of the termination of their vertical migration, you can see that they normally stop or terminate their vertical migration at a nitrate concentration around between 3 and 8 micromolar. So that's what I mean by the vertical migration is initiated by light, which means that they start getting down close to the fat side, and then they stop at a certain concentration of nitrate concentration. And the next thing I want to point out is that based on this slope of this vertical migration patterns, we are able to estimate the group velocity or the because we are measuring them as a group. So basically the phase velocity of this vertical migration of this algal vertical migration. So which gives us the value around 400 microns per second. So in order to better understand this value, this is actually 10 body lengths per second for them. And then they are able to swim for a duration of like 18 hours to 24 hours. So which is far more beyond the ability of our human beings. Like for the Olympic swimmers, we may swim like 2 body lengths per second, like with a maximum like 1 hour most. But they are able to swim like 10 body lengths per second with a duration of like 18 hours or 24 hours, which is just an incredible swimmers for me too. I'm so impressed by this. Yeah, it's so amazing. And then the first thing is that you can see this, the last panel is nitrate concentration. You can clearly see a lot of nitrate concentration, especially during this first several days of the measurement. So you can see that along this isopycnal, there's a clear decrease of nitrate concentration on this isopycnal. So we be wondering like what is the reason for this loss of nitrate? Were they lost because of the assimilation by algal, or were they lost by some other like physical advection mechanism? For example, some low nitric water mass was advected into our circular region. So in order to understand this reason, so the next thing that we do is to quantify what is the biological assimilation rate of nitrate by the algal using this nitrate data. So then. But before I get into too much details, I just want to show this nitrate loss, which is what normally people understand as the temperature nitrate relationship. Basically, you can think about that. So what is the nitrate concentration on each specific atmosphere? So I'm plotting two different lines. So basically, the nitrate temperature relationship based on 70 years of climatological data are plotted in purple. So this is a very nice linearly purple line here. And then the nitrate temperature relationship during this experiment, or in other words, during this red tag, was plotted in green. So basically, you can see that there's a clear this triangle region where there was a loss of nitrate on these isosurfaces. So in order to better understand, so this region between like 14 degrees C isosurface to around 11 degrees C isosurface has a vertical coverage, a vertical extent of 30 meters, and then the loss of nitrate is up to 13 micromolar. So this is a huge loss of nitrate during this red tide event compared to the climatological situation. Then in order to understand what is the mechanism of this loss of nitrate, so what we did again, and I mentioned, is to quantify this nitrate uptake rate. So this slide is a little bit too much about technical stuff, but I want to just go through this real quick. So what we did is that the one difficulty is that because we are aware of the advection of different water masses, especially by the waves during this red tide event. So therefore, the question is how to decouple this horizontal direction of the internal waves with the biological uptake of nitrate. So so Peter, especially Peter, developed this very smart way of decoupling these two effects by using spiciness as a tracer to follow the same water mass as a function of time and also space. So for people who are not familiar with spiciness, so therefore, so think about this as along the same isopycnal. So you can have both cold and saltier, cold and fresh water, and also warm and saltier waters. So these two different waters, they are, even though they have the same density, but actually they are different water masses, and they may come from different origins. So this spiciness is actually a really great tool to try to differentiate these water masses on the same isopycnal. So in other words, the cold and the fresh water can be called as mean tier, and then the warm and salty water, they can sometimes called a spice tier. So here I'm showing the temperature salinity plot of data on a specific isopycnal. So therefore, each data set were on the same isopycnal, but the dashed line represents the spiciness beam that we are able to characterize different water masses. So therefore, with this spiciness, we are able to trace, because we believe that this TS property is able to be conservative, independent of the direction of the water mass. So therefore, the nitrate change inside each spiciness beam is believed to be purely or primarily due to the uptake of nitrate inside this water mass. So with this adoption, and also therefore we are able to quantify the nitrate loss within each spiciness beam as a function of time, so which are all the lines here. And then based on this decaying of nitrate within each spiciness beam, we are able to quantify the nitrate uptake rate, or in other words, nitrate loss rate, and also map this nitrate loss rate to the back in time to estimate what is the temporal and spatial map of the nitrate loss that is only due to the biological uptake. So we are aware that this method is not the best, but that's actually the best approach that we are able to get. And also we just want to show some PDFs of the nitrate uptake rate and compare this rate with the lab-based measurement rate from the previous literature, and they are really good. There are really good agreement with each other, which indicating that this spiciness focus nitrate uptake rate is durable to give us some reliable estimation of the nitrate uptake rate during this rectangle event. And then with this technology, we are able to estimate what is the nitrate loss that is only due to the biological uptake, which is what I'm showing here in this upper panel. So again, I'm showing the estimated loss of nitrate, which is only due to biological uptake in these contour lines as a function of isopycnal and also time. And then, the contour lines is the fill of the nitrate anomaly, which is only based on the measured nitrate. And then the color is the estimated biological loss of nitrate. So you can see that they are really good correspondence to each other, not only in the regions, but also in the magnitude. And then another thing that we did is that we are able to do a depth integration of this loss of nitrate that is only due to the biological uptake, and which is plotted in the blue line here. And on top of that, we also plot the depth integrated chlorophyll concentration and also octability, which is optical backscatter data in black and red respectively, which are these two lines here. So you can see that there's a really clear temporal correspondence between the increase of the chlorophyll biomass with the loss of nitrate as a function of time. So again, with this evidence, we believe that. So the story is that as nitrate is gradually being assimilated, the biomass gradually increases, which is a direct evidence to show that this dinoflagellate or this phytoplankton, they are able to assimilate nitrate and convert this assimilated nitrate into biomass and then grow. So back to the last slide I want to show. So with all this evidence combined, so we are able to show a story that this dinoflagellate, they are able to swim from the surface to the deep nutrient pool once the sun goes down. And then once they were in the deep nutrient pool, they assimilate nitrate and then convert them into biomass. And then that's at least what we think is a direct justification of this diurnal vertical migration nitrate hypothesis that has been brought up of over half a century ago. So again, with a 4 points of summary. So first thing is that we are able to show that this polyp, they vertically migrate at night and takes up nitrate from below the floating zone. And the second thing is that the migration is initiated at sunset, and the migration depth actually depends on the deep nitrate concentration. And then the third thing is that the understanding of the physiological and also environmental drivers of dinoflagellate swimming behavior will be crucial to enhance our ability to focus the HABs. And then the last point is that our data provide an excellent platform for building models of dinoflagellate behavior interacting with physical and environmental forces to create HABs. So I think there's like one last point that I really want to emphasize on is the combination of different technologies that really enables us to measure both. For example, the FCB gives us critical information on the macro environment or micro components of the red tag, and then the wild data provides us a great opportunity to understand the physical and general background, physical and biogeochemical environment of these macro organisms are living in. So again, this combination of technologies from different aspects is really powerful to give you a full picture of what was happening subsurface and then especially during this red tag. But before, I think I still have some time left. So, but before I end my talk and answer any question, I just want to share a little bit about my current work, which I'm doing at HUI right now. So basically, I'm under this project called NES LGER project, which is Northeast Shelf long-term ecological research. So basically, I'm showing the map. So basically, that here with a star here, with a heart here, and this is the black boxes or like our sampling region. So where we go out like 4 times a year every season to collect this transect from this is the Cape Cod, from the Massa's Vineyard, which is here, down out to the outer shelf rig to collect collect the physical profiles and also biogeochemical profiles and also micro like growth rate and also the phytoplankton concentration and also their primary production rate at targeted stations like 4 times a year, and try to understand what is the ecological rule or ecological change of this region, especially in response to the change of the climate. Because this region is not only important in the physical dynamics, because that's the place where the Gulf Stream and also the shelf water interact with each other, but also because of their economical interest, because this is also a place famous for a lot of fishing and all these economical impacts. So in terms of my research, I mainly studied the nitrate dynamics again, which is this panel here. So here I'm showing like 5 different panels of the cross, only an example of the cross-shelf distribution of different oceanic parameters as a function of depth and then also cross-shelf distance. So as I can see, temperature, salinity, density, chlorophyll concentration, and nitrate. So basically, I'm trying to understand how the nitrate is able to evolve in response to different physical dynamics and also the micro activities. So again, like I'm able to use this ball of 5 years of data to characterize what is the mean nitrate profile over this northeast continental shelf and understand what is the controlling mechanism of different portions. I can see there are like clearly 3 different portions of the subsurface nitrate profiles, and I'm trying to understand what is the constraining mechanism that is able to characterize this nitrate profile. And also besides this, I'm also trying to understand what is the export rate, or in other words, how much organic materials is able to be exported from the surface euphoric depth zone to the subsurface. And then that's because that's a really huge consequence, not only regarding this ongoing climate change, but also carbon sequencing and all this stuff. So there are 3 sub-questions that I want to understand. For example, the first, as I mentioned, is to understand what is the biophysical constraint on the subsurface nitrate profile. And also the second one is what is the ecological rule of this vertical nitrate flux regarding to the local permittivity. And the third thing is to get a better understanding of the 3-dimensional, for example, the 3-dimensional nitrate flux that flux into this sampling region, and then quantify what is the seasonality and also the temporal evolution of this flux into the local system. So yeah, with that, I would like to back to this summary plot and then answer any questions. And thank you for your attention. Yes.
So thank you for a very interesting talk. I'm intrigued by the swimming speed that you talked about. Have you calculated the amount of energy they need to sustain that swimming and compared it to the photosynthetic carbon fixation rate?
That's a really good point. We haven't done that, but that will be an interesting approach.
Yeah.
Because it definitely took them a lot of energy to be able to swim down deep and also swim with such long duration. And then another thing that I really want to I want to expand this a little bit is that we have been wondering like what is the controlling, like why they didn't dive even further deeper down. And then that's one of the hypotheses that they may not have enough energy to be able to sustain them much longer from the surface, like without any new energy input. So that's again, so this calculation of the energy could be a really important evidence.
A very simple calculation to make.
Yeah.
Calories
Yes.
expended per hour or something.
Exactly. And then that's a really good point. And then to at least provide some evidence of what is controlling the maximum depths that they are able to work with. Thank you. Yes.
Good job.
Yes.
So thanks very much for a great talk.
Yes.
There's other phytoplankton in the sea. And I wonder whether things like storms and tides allow other species in the California bight to take over these poor, sad dinoflagellates that have to swim up and down. There are other things that if there's turbulence, are there other things that just take over?
That's a really good point. I think especially during the winter time when there were like a lot of storms, and especially the springtime when there was like enhanced coastal upwellings, which brings like a lot of nutrients from the subsurface into the surface, we will see like the water column is mainly dominated by diatoms, which is larger cells than the dinoflagellate. And then so that's yes. So with the storms, so with this input of the new nitrate or new nutrient from the subsurface, you are able to see a change of the species at this local Southern California region. And then one of the reasons that this this dinoflagellate is able to dominate during this red tide, at least what we think is that because, as I mentioned, the water column is strongly stratified during this time period of the year, because it was an enormously warm year during this April and May compared to the other years. So this whole water column was stratified, and then the diatoms there. And the first thing is that with this stratification, you have limited nitrate flux from the subsurface to the surface, so which is not really good. At least these diatoms, they are not happy with this environment. Another thing is that these dinoflagellate, they really like these stratifications, because this vertical migration or swimming ability is able to let them overcome the diatoms in terms of competing for nitrate. So that's, I think, another, at least for this red tide, for this stratified situation, you would expect a more like a dinoflagellate dominate subsurface micro community in the Southern California.
But it's a really special condition that allows this to happen.
I think so. Yeah, it's
and it's not a normal thing. Normally, there's turbulence.
Normally there will be turbulence. Yes, that as I mentioned, yeah, this is not a normal situation, but this is actually happening more and more frequently regarding to the changing of the climate. So
Why is that?
Because you have the water is getting warmer and warmer, especially with increased stratifications. So therefore, at least normally we would expect this happen like once every several years. But this and also it's mainly during the springtime. But this year, like this air quality, like just caused the red tide in Southern California back to August or September. So which is also an abnormal situation, and then it's coming like more and more frequently compared with other like previous years. Yes.
So I'm not entirely sure how nitrate concentrations fluctuate with changes in the magnitude of upwelling, but assuming that this was done during a fairly strong La Nina phase. How do you think, if you think the way that vertical migration of dinoflagellates would function this way, how do you think this would change in magnitude in the newer El Nino phase that we are currently in, if it would change, and if so, I wonder, would then this vertical migration be deeper? Would they have to go further down to get to their nitrate, or?
Yeah, that's a really interesting point. I'm not too sure, but there could be some connections of like, especially modeling work, I'm able to see how this vertical migration behavior is able to respond or react to this changing of the larger scale climate forces. But that could be, yeah, I think that could be, yeah. Yes.
Yeah. When you look at the downward migration, it's stronger than the upward. Can you explain that?
Yes, that's a really, really great observation. So we noticed that because exactly as you can see, this downward migration is more organized or coherent, but the upward migration is not as coherent as the downward migration. I believe that's. And then we didn't dive too much into this. And then there are again, like we have some discussions, especially hypotheses on this less coherent upward migration. So one hypothesis is actually date back to Peter's work, which is all modeling work regarding this upward migration is triggered not by multiple things. For example, this downward migration we believe is triggered by light, but this upward migration may be triggered by, for example, nitrate concentration may be triggered by other environmental factors. Therefore, they are asynchronized regarding their upward migration, because some cells they may met their upward migration criteria and then they swim up, and some other cells they may not have met their upward migration criteria and they will start diving down. So in other words, they are so we see a very synchronized downward migration instead of the asynchronized upward migration. So that's one hypothesis. The other hypothesis is that, so this one thing that I didn't mention is that this alcoholic, especially most of the dinoflagellates, they are mixed trophic. So which means that they are not only they are able to gain energy not only by photosynthesis, but also eating like preying smaller cells that cells that are smaller than those. So therefore the hypothesis is that they may not need to come back to the surface that urgently. So therefore they can stay subsurface for some time and prey on other stuff and gain energy. So therefore again the thing is that they don't have to come up to the surface as coherently as a group. So that's again, it's a really interesting observation, and we are not too sure about that. And then we have some hypotheses and that can be tried in the future to expand on this. But thank you for the question. Yes. Yes.
I have questions. What's the nitrate concentration level at which dinoflagellates would like to first migrate to, and why?
That's a good point.
Yeah, from your slides, it's around 8.8 micromole.
Yeah.
So why is that the concentration?
That's I don't. So that I mentioned this point is that they seem to terminate their vertical migration at this nitrate concentration around 3 or 8 micromolar. So it seems to be they are a preferable nitrate concentration. And then I'm not too sure, but it's also very surprising to me that they seem to have some kind of nitrate sensor. So once they sense that they are at some environmental conditions with a nitrate concentration around this like 0.8, they know that, oh, I'm in this nutrient pool. So therefore I don't have to dive down even deeper. So yeah, it is pretty smart to be able to sense the environmental nitrate concentration and then determine their physiological behavior based on this environmental nitrate concentration. So, but in terms of the actual preferable nitrate concentration of this species, I'm not too sure about that.
What's the
Sorry? Yeah,
Total water depth.
the total water depth is a 100-meter depth. But I'm only showing the upper 50 meters. Yeah. And then again, this is sorry. Yeah. And
That's
again,
your answer.
no, I just want to this is like 30 meters is far below the euphoric depth. But, yeah, that's another evidence. Yeah.
So, you didn't mention whether there are any other forms of nitrogen here other than nitrates, and you did talked about mixotropy, and I'm wondering if you went back and went to your original question about stoichiometry of how you're supporting any given carbon to nitrogen ratio in the primary production taking place.
That's a good point. That's true. We do not have other form of nitrogen measured during this red hat event. But I didn't show the evidence, but we did calculate what is the nitrogen based on nitrate to carbon ratio from this plot, which is very simple. You can just do a ratio between between the black line with the blue line to calculate that ratio. And then that ratio, we did compare that ratio with the literature-based carbon to especially carbon to chlorophyll ratio for alcoholic, and they are reasonable and in very good consistency with each other, which is indicating that the nitrate is a predominant source of nitrogen for alcoholic during this time.
And I'm sorry, but do they have a particular highly fixed carbon to chlorophyll A ratio throughout this entire growth process?
I think it's a carbon to chlorophyll ratio around 1 to 20. I can't remember exactly. But I can take that deeper. It's in my computer, and I actually remember. But again, the point is that they are consistent with the literature. But again, the literature is not based on the red tide. So therefore, which means that during the red tide, the carbon to chlorophyll ratio of this species is sort of consistent with their carbon to chlorophyll ratio during their normal situations. I don't know whether I understand your question.
No, that's fine.
Okay, yeah, thank you. Yeah.
All right, if there are no more questions, then thank you so much for this really interesting talk.
Thank you. Yeah.